Dear students, I hope all of you are doing well. Today we are going to discuss classification of viruses on the basis of structure. As we know, there are two important components of virus. First is the protein coat called as capsid. Second one is the nucleic acid called as genome. First we will classify viruses on the basis of shape of capsid. Some viruses have helical capsid, for example, tobacco mosaic virus. Some have polyhedral capsid, for example, adenovirus. Enveloped virus have host cell membrane covering outside of capsid, for example, influenza virus, and some viruses are naked. Some viruses have complex. Now we will classify viruses on the basis of type of nucleic acid present in their capsids. Some are double-stranded DNA viruses. For example, smallpox virus, adenoviruses, herpes viruses, and hepatitis B virus. Some are single-stranded DNA viruses. For example, parvovirus, which cause mild rash, and bacteriophage. Some viruses have double-stranded RNA molecules. For example, reoviruses, which cause diarrhea. Some are positive-sense single-stranded RNA viruses. Positive-sense RNA is that which can act as a mRNA for protein synthesis. For example, rubella virus, hepatitis A and C virus, polio virus. Now, some are negative-sense single-stranded RNA viruses. Negative-sense RNA is that which cannot act as a mRNA, but used as a template for mRNA synthesis to manufacture protein. For example, influenza virus, paramyxovirus, measles and mumps, and rabies. Some viruses have diploid single-stranded RNA, mean having two single-stranded RNA molecules, which act as a template for DNA synthesis by a process called reverse transcription. For example, retroviruses, HIV.